Protein synthesis is a fundamental process in all organisms and cells that involves 5 stages: activation of amino acids, initiation of translation, elongation, termination, and protein folding with post-transitional processing. These steps occur in ribosomes, which are key components to protein synthesis. Without proteins, many cells wouldn't be able to carry out their day-to-day functions at all. A lot of antibiotics utilize this fact and target protein synthesis in bacteria to prevent their function. Antibiotics are one of the most commonly known and beneficial medicines and are used to fight bacterial infections. Different types of antibiotics work in different ways. However, the type of antibiotics that we are going to focus on are protein synthesis inhibitors. Ribosomes are the prime target of these antibiotics because they are the sites of protein synthesis. One issue that arises when targeting ribosomes is that all cells contain them, both bacterial and eukaryotic, and antibiotics only need to target the bacterial ribosomes. Luckily, the ribosomes in bacteria and eukaryotes are structurally different from each other. Bacteria have 70S ribosomes, while eukaryotes have 80S ribosomes, which have different molecular weights and shapes. This allows antibiotics to specifically target bacterial ribosomes. All ribosomes are composed of different subunits, and bacterial ribosomes have 2 subunits, the 50S and the 30S. These subunits play different roles in the process of protein synthesis. Before discussing more about the specifics of how antibiotics inhibit protein synthesis, let's review the normal process of protein synthesis and the role of the ribosomes in bacteria. First, the initiation complex is formed by combining the 30S and 50S subunits and binding of the initial amino acyl tRNA at the P site, or peptidyl site. Then, a second tRNA with a particular amino acid binds to the ribosome at the A site, or amino acyl site. The anticodon on this tRNA matches the codon of the mRNA. A peptide bond then forms at the P site between the old amino acid and the new amino acid. The bonded amino acids then shift to the E and P sites. The empty tRNA then exits at the E site, or exit site. This process continues as the polypeptide chain is elongated until it reaches a termination sequence and the components dissociate. There are many different ways that antibiotics can interact with ribosomes to inhibit bacteria protein synthesis and therefore cellular function, because the ribosome is involved in all steps of protein synthesis. One class of antibiotics that inhibits ribosomes that we are going to focus on is tetracyclines. Tetracyclines are a group of broad-spectrum antibiotic compounds that have common structure and exhibit activity on a wide range of microorganisms. They are all composed of a linear fused tetracycle nucleus with a variety of functional groups depending on the specific type. These functional groups don't change their antibacterial activity, but rather affect pharmacological properties. Tetracyclines are used to treat bacterial infections of the skin, intestines, respiratory tract, lymph nodes, and other body systems. It is most commonly used to treat acne as well as sexually transmitted diseases such as syphilis, gonorrhea, or chlamydia. Tetracyclines have a very specific mechanism of action that inhibit protein synthesis at the initiation of translation. They bind to the A site of the 30S subunit, which prevents the binding of amino acyl tRNA to the A site on the mRNA. This inhibits the elongation of the polypeptide chain because the next bead of the polypeptide string cannot be brought into the ribosome because the site is already inhabited. This prevents the following stages of translation and therefore the production of proteins. Without proteins, the bacterial cells die off. Tetracyclines are also effective because they simultaneously inhibit other steps of protein synthesis, which causes the death of the cells to occur faster and helps prevent the antibiotics being ineffective. Promoting protein synthesis is an integral part of the ability for all organisms to function, yet the inhibition of protein synthesis has also saved millions of lives.